Welcome to Biotech and Science Channel.
Let's talk about ribosomes.
Definition. Ribosomes are cellular structures responsible for protein synthesis, functioning as the machinery that translates genetic instructions from RNA into proteins. These tiny organelles, found either floating freely in the cytoplasm or attached to the endoplasmic reticulum, consist of two subunits, small and large, that come together during protein synthesis. Key points about ribosomes. 1. Protein synthesis. They read messenger RNA, mRNA, transcripts and translate the genetic code carried by mRNA into specific sequences of amino acids, forming proteins. 2. Two subunits. Ribosomes are composed of a small and a large subunit, each with their specific roles during translation. The small subunit reads the mRNA, while the large subunit joins amino acids to form a polypeptide chain. 3. Location. They can be found in two main cellular locations, free-floating in the cytoplasm or attached to the rough endoplasmic reticulum, ER. Ribosomes attached to the endoplasmic reticulum are involved in synthesizing proteins that are either secreted from the cell or incorporated into the cell membrane. 4. Conservation of structure. Despite variations in cells, the basic structure and function of ribosomes are highly conserved across species, showcasing their fundamental role in life processes. 5. Ribosomal RNA, rRNA. Alongside proteins, ribosomes contain rRNA, which plays a critical role in catalyzing the formation of peptide bonds between amino acids during protein synthesis. 7. Ribosome composition. Ribosomes are made up of two major components, ribosomal RNA, rRNA, and proteins. The small and large subunits consist of a combination of ribosomal RNA molecules and various ribosomal proteins. 8. Assembly. Ribosomes are assembled in the nucleolus, a distinct region within the cell's nucleus. The assembly involves the synthesis of rRNA, which combines with ribosomal proteins to form the small and large subunits. 9. 3 stages of translation. Translation, the process carried out by ribosomes, occurs in three main stages: initiation, elongation, and termination. Each stage involves specific interactions between mRNA, transfer RNA, tRNA, ribosomal subunits, and amino acids to build the protein chain. 10 types of ribosomes. Eukaryotic cells possess cytoplasmic ribosomes for protein synthesis within the cell and also have ribosomes associated with the rough endoplasmic reticulum, RER, involved in synthesizing proteins for secretion or membrane-bound proteins. Synthesis. The number and activity of ribosomes in a cell can be regulated based on the cell's needs for protein synthesis, adjusting to changing environmental conditions or developmental stages.
Thanks for watching. Don't forget to like, share and subscribe.